Hey everybody, good afternoon. On behalf of Fight CRC, Sharon and Andy are joining you. We actually have the distinct pleasure to talk to and introduce Dr. Kevin O'Rourke, who is going to be joining us to talk a little bit about his work as an emerging physician and research scientist. We actually were introduced to Kevin from Dana, one of our board members in New York. And so we're really excited to hear from Dr. O'Rourke about all of the work that he's doing. And I understand he just defended his thesis only a few short days ago, or actually about a month ago. So we're excited to hear from Dr. O'Rourke about the work, talking a little bit about how this fits into the overall schematic for research science. And one of the fun and interesting things about Kevin is that he had, with all of the work and advancement that he's made, he's been noted as one of Forbes 30 under 30. So we were really excited about that honor of distinction and having that sort of elevated status, particularly around a young scientist who's doing work in the area of colorectal cancer. So super excited about Kevin's work, bringing that today as part of discussion around research advocacy. We're going to have time for Kevin's discussion, presentation, and then open up the line, I believe, for people to ask questions and to really hopefully open a dialogue. I think we can do that, but Sharon will give us instructions about exactly what to do. I do know that during the course of this presentation, you can key any specific questions that you might have. So we're really excited about having Kevin on. And a huge thanks to Dana, our board member, for bringing him to us to make this sort of fantastic connection so that we can help really grow the research advocacy work and have all of you really aware of the great things that are happening in the field. So Sharon, I'll punt to you for any other details, comments you want to make, and then we'll get started with Kevin's presentation.
Sure. So my only comment really is about the questions. And I know most of you have been on the more traditional Fight CRC webinars. So similar to that, you can type in your questions on the right side of your panel so that you don't lose track. I'll double check and see if we can open up the line so you can, we can have more of a dialogue today. But if not, then certainly make sure that you're typing in your questions so you don't lose track. And that's it. So Kevin, thank you again, Dr. O'Rourke, and I'll let you take it from here.
Okay, thanks everyone. One quick question about how long do you want the slides to go for before we finish and wrap up our questions?
You know, Kevin, I think if you could go for probably 25 or 30 minutes, that would be plenty. And then we can give time for people to wrap it up. And if there's something you want to delve into, I think, Sharon, we have an entire hour dedicated to the discussion or to the discussion and Q&A. Is that right?
Yes.
Okay, so I think if we could aim for at least 30 minutes of content, and then Kevin, if there's more that you want to delve into, by all means, please shoot. But we'll open up the lines. We probably want to leave at least 10 or 15 minutes for any questions that should come up. Okay,
fantastic. And if any questions come up and there's a method to ask them during the presentation, I'm happy to field them as we go along too. It doesn't have to be completely uninterrupted. So.
Perfect. Sounds great.
So thank you, everyone. This has been a really cool chance to contribute to Fight CRC. As it was kind of mentioned, I met Dana. We actually both attended the same undergraduate institution together, but didn't really connect until a mutual friend said, "Hey, Dana, Kevin's working in colon cancer at Memorial Sloan Kettering Cancer Center, and you guys should connect." And since then, I've had a chance to kind of meet the staff, and it's been a really positive interaction. So I'm just as excited as you are to be here, and I thank you for the opportunity to talk. These slides are the exact same slides that I presented to the faculty at Weill Cornell Medical College and Weill Cornell Medicine, which includes their hospitals, as well as the folks at Memorial Sloan Kettering Cancer Center about a month ago. And at the conclusion of which they granted me the PhD. So you guys are getting the real deal here. But I'll kind of go through some of them faster than others because perhaps it's not all relevant. But I will start off with a kind of broad overview of where we are in the field and what needs to be answered in order to make advances. And you know, the main focus of my work, as the title says, has been to develop new models of colon cancer in order to facilitate new therapies for metastatic disease. And I know this is a learned audience, but I'll go into exactly what that means right now. So let me just see if I can advance. There we go. Great. So the first thing I think that I learned in the 4 years of doing a PhD on this topic was that the intestine is a remarkable organ. I'm not so sure I would have said that before I started, but it really truly is incredible when you add up all of the things it does and how it accomplishes it. So the small and large intestine are part of your gastrointestinal tract, and their job is to absorb water, minerals, sugar, fat, proteins, and vitamins from the food you eat. And obviously this is required for life. To look at the structure of how that works, we'll just kind of, you know, take a cross section of the small intestine, which is to our left. And we know that, you know, lumen or the center of the intestine is where food and bacteria live. In fact, there's almost or you know, thought to be hundreds of species of bacteria that live in an ecosystem, you know, in this human microbiome that reside inside the organ. So this is the first kind of unique thing about the organ, in that the microenvironment is not just human cells. There's a very, very large, diverse jungle, if you will, of viruses and bacteria and things that reside in the organ. And that's important to take into account, especially down the road whenever we think about studying that organ, right? And then the layer of the intestine that does majority of the work is called your mucosa or epithelium. It is in contact with the outside world, just like your skin, right? It's being exposed to the things you eat, anything you swallow. Underneath the mucosa is a layer called the submucosa, and underneath the submucosa is a layer of muscle. And finally, in all of these layers are a network of lymphatic and blood vessels. You can kind of think of them like a little, you know, like weeds that kind of spread out throughout all these layers to take the things that you absorb from the lumen and then distribute them to your body. Now, what's impressive is that the mucosa is actually the size, when you look at the small and large intestine combined and you were to lay the mucosa flat, it'd be the size of a tennis court, which is a pretty large surface area. And then on top of that, what's even more impressive is that the body replaces that entire tennis court every 4 or 5 days. And so to examine that process, we'll look at it a little bit closer. We call that shedding and replacement of cells stem cell homeostasis. And so here what we're looking at, I'm not sure, can you guys see my pointer on the screen or no?
Yes.
You can, okay. So this right here is a layer of mucosa in the human colon. And you can see that there's these architectural units called crypts. And at the base of crypts are stem cells. And their job is twofold. They must divide throughout the course of our lifetime, throughout adulthood. And then their daughters go on to, the daughters of cell division go on to migrate up the crypt and continue to divide to repopulate the mucosa. And as they kind of head up this conveyor belt, they differentiate, right? So they turn into cells that are there to accomplish the purposes of the organ. In this case, it's to make cells that absorb salts and sugars. Those are called absorptive enterocytes. There are these large kind of vacuolar cells that contain mucin, and that mucus is secreted to allow the smooth passage of the contents of your intestines. And so as that process happens, these cells migrate up and eventually become shed and excreted with your waste. And so this kind of conveyor belt is constantly in flux, sending new cells up the belt and old cells out. And here's just a kind of a cartoon diagram of one of those crypts. Again, we've got these stem cells. Their daughters head up the crypt, divide, differentiate into different cells whose job is to accomplish what we already talked about, and then eventually they're shed. So this brings us to colon cancer, because essentially when that process goes wrong, we have the disease called colon cancer, and this is the second leading cause of cancer death in the United States. And according to the estimates, there'll be 135,000 new cases this year, resulting in 50,000 deaths, and about 1 in 20 people will be affected. Now, we kind of reviewed what the normal colon looks like, and there's a couple key features of colon cancer that are almost immediately observable when you look at what the normal colon looks like as it transforms into colon cancer. So the first thing that we kind of look at, we see is like a bomb went off, right? This is colon cancer. And so the features are that the cells that normally underwent stem cell homeostasis, these are normal cells here that are undergoing that shedding. That shedding is no longer occurring, right? So whereas the cells would normally not to be dividing, we have cells that are dividing. The next is that orderly mucosal lining here that has beautiful crypts is now completely lost, and that architecture is lost. And finally, and the hallmark of cancer are malignant invasions. Now the mucosal cells are dividing into the submucosa and eventually the muscle layer. And if left unchecked, they'll grow into lymphatic systems and eventually other organs. And that is the hallmark of the progression through disease. So that's important because this stepwise progression from a stage 0 colon polyp or adenoma into a frank cancer where cells are invading the submucosa, eventually into a stage 2 tumor that's invaded the muscle, finally to next to a stage 3 tumor that goes into the lymphatic system, and finally to a stage 4 that is spread to other organs, is a hallmark of colon cancer. And this progression is important because we know that survival can be dependent on the stage of the tumor at which the patient is diagnosed. So if we look at patients who are diagnosed at time 0, this would be like when they come to the clinic and they're diagnosed with stage 1 colon cancer. 5 years later, the survival rate is above 90%. In most cases, it approaches 98 or 99%. What's troubling though is that if we catch the tumors later at say stage 2 or stage 3, whether or not a patient is still alive 5 years later, depending on the treatment they get and all that, is about 50%. And so here comes our first question in colon cancer research, is if I have a patient at time 0 upon diagnosis and they have a stage 2 or 3 colon cancer, I would like to know now at time 0 whether or not they're going to be part of the 50% that will respond to treatment and still be alive 5 years later, or if the treatment that I'm going to offer as a clinician will not be effective and they will not survive the disease 5 years later. And so that understanding what biomarkers might be available, or these would be like clues that we could look at in either blood testing or fecal sampling or things like that, that might tell us how this tumor will eventually develop 5 years later. And then the most troubling statistics are that when you diagnose a patient with stage 4 colon cancer at year 0, survival is dismal. It can be less than 10% at 5 years after diagnosis. And so this also tells us another research question, which is that there must be something inherently different between a stage 1 colon cancer and a stage 4 colon cancer, and even a stage 2 or 3 colon cancer and a stage 4 colon cancer. And so you really need, in order to study the differences between stage 1, stage 2 and 3, and stage 4 colon cancers, is a model that can recapitulate all those 4 stages, right? And again, about 1 in 5 patients who come to the clinic are presenting with metastatic disease, and they're bound for this survival rate of less than 10%, which means we need to put in work to identify new therapies that can alleviate and cure or reverse cancers that are diagnosed at stage 4. And so how has the field tried to address this? Initially, or not initially, but one step is to simply reduce the risk of getting colon cancer in the first place, right? And so, for example, there have been successful studies that have shown that a group of drug called NSAIDs, which include like aspirin or others, are able to reduce the risk of adenomas or even adenocarcinomas in patients that are at risk for those colon polyps. The other method that's been probably the most successful at reducing deaths from disease is to detect it early, right? So if you can detect a stage 1 colon cancer, a surgeon can resect it, and the patient's cured in most cases. And so the trick is routine colonoscopy. And I know that the Fight CRC folks have done an amazing job in increasing outreach and awareness to people to get routine colonoscopy. And this, I think, is recommended for patients over the age of 50 or earlier, depending on family history or things like that. And finally, we need to develop new therapies. And so this is a Time magazine article that's, I think, 15 or 16 years old, about a new drug called Gleevec, which was designed after we learned exactly what cured, or actually exactly what caused a very specific type of leukemia. And so when you understand the exact genetic mechanisms that drive that leukemia, that the doctors were able to engineer a therapy that was targeted directly at those genetic alterations. And as a result, Gleevec took a disease that had no cure and completely reversed it. And now it's very manageable. So chronic myeloid leukemia used to be a death sentence in the 90s, and now it's a curable chronic condition, right? And so that's the thought. That's the biggest, holy grail example in the field of where, if you can study and understand what drives colon cancer, maybe we can engineer therapies to cure it. And so to do that, what you have to first do is understand what drives colon cancer. And the place to start is to ask, okay, well, what's mutated? What genes are commonly mutated in the disease? So you start with human colon cancer specimens and you ask, what genes are mutated in colon cancer? Now, as you do that, you find out that there's, on average, though there can be, 30 genes or 100 genes or sometimes even thousands of genes mutated in a single patient's colon cancer. And it's unlikely that every single one of those 1,000 genes is actually the driving cause of the disease. And so the question is, of the most recurrently mutated genes in, say, 1,000 different patients' colon cancers, which ones are the actual genes that are causing the disease? And so to answer that, the gold standard method in colon cancer research has been to introduce those genetic mutations into mice. And then you examine the mice for colon tumors, right? So it's a pretty simple test. If you thought that a gene in humans was causing colon cancer, and you give that exact mutation to a mouse, and they go on to develop a colon tumor in their colon, then you've got pretty good evidence, right, that that gene is causing the disease. But this actually then enables you to do more than just call that gene a driver, because now you've got a mouse that reliably gets colon tumors, right? And so you can use that mouse to say, well, are there new therapies that we could administer to the mouse that would be safe and effective at slowing that disease? Or you could ask, are there biomarkers? Are there things in the blood of this mouse that are telling us what the stage of the disease is, or how it will respond to therapy? Or you could even imagine that the mouse would serve as a method to detect new ways to detect cancer at all. So our gold standard is colonoscopy, but if there were a cheap, ubiquitously available at-home kit that patients and could use, that might solve the problem of not being able to get colonoscopies to areas that may not have a clinic within an hour drive, for example, or things like that. And so the first attempt to understand that kind of paradigm really began in the 80s and kind of culminated with this landmark paper 30 years ago by Fearon and Vogelstein. And I know you can't read this, so I'll show you an updated version, but the point is that in the 1990s, researchers kind of looked at that stepwise progression from adenoma to adenocarcinoma to metastasis, and they asked what genes were currently mutated, and they found a pattern. And it kind of looks like this. So the initial gene that's thought to be disrupted that converts that normal mucosa into that uncontrollable cell division with the loss of tissue architecture is a mutation in a gene called APC. And APC is a tumor suppressor, so its normal job is to block this from happening, but when its function is lost, then you get the conversion to stage 0 colon tumor. And then it's the stepwise accumulation of additional mutations in other genes that leads to that malignant invasion and eventually full-blown stage 1 colon cancer and eventually stage 2, 3, and 4. Now I make a quick joke here. So the question is, it's been 30 years since we learned this. What have we done with it? And the joke is, what you do is you put a bunch of medical students in a room and you make them memorize it. So this is a question from a board exam. And in order to be a physician in the United States, you need to know what I just told you all, which is that normal epithelium to early adenomas in humans is thought to be caused by the loss of the gene APC, and then the next gene is KRAS, and the next gene is p53. So we've known this so long that doctors are required to know it, but we still don't have any smoking guns as far as what exactly is the most important gene in colon cancer. So that kind of sets the stage for where we are in 2017, and we view genetics of disease with these plots that I'll kind of explain to you. So we have genes on our left, APC, we've kind of introduced that one already, and 3 others. We'll call them SMAD4, p53, and KRAS. And to imagine a single patient's tumor, we'll put them in a column. And if they have a mutation in APC, we'll give them a little green box. And if they are like normal for that gene, it'll be gray. And so we can kind of look at colon cancer as a whole by saying, okay, well, let's look at a couple dozen patients. And the first picture that emerges is that the genetic combinations of just these 4 genes in a patient population of say 2 dozen or so here is heterogeneous, or it's mixed. So some patients have mutations in APC and SMAD4, some have it in APC and KRAS, and some actually don't have mutations in any 4 of these genes at all, right? And these are actual patients that were sequenced here in the past year at Sloan Kettering Cancer Center. But of course, we know that there are hundreds of genes mutated, and we've now sequenced thousands of patients. So we've repeated these analyses for hundreds of genes and for lots of patients, and the picture becomes a lot more complicated. So now we're viewing 1,500 or 1,500 patients at Sloan Kettering Cancer Center for just those 3 genes, APC, KRAS, and p53. And the columns are now so skinny they appear as very, very thin lines, right? And the situation gets really kind of dire or complex to understand, because when we look at the next most commonly mutated genes in the disease, what we see is that there's a very large mixture of the genetics of what a single patient can present with. And so in total, what I've just told you in one sentence is that no 2 patients are the same genetically, right? So this is thought to be the reason why we see heterogeneity with response to treatment. So if you take 2, for example, stage 3 colon cancer patients, one may have a really good response and one may not, and we don't know why that is. And it's thought because maybe they have some kind of mutation in PTPRS that, in combination with APC and KRAS, for example, is going to make them resistant to the current chemotherapies we have available. And when you add up all the possibilities that could be explaining the resistance that we're seeing to the current available treatments, it becomes like a very large task to try and understand this snapshot, right? And so the kind of underlying the question that has driven a lot of colon cancer research is to find if there are any genetic commonalities that are driving colon cancer. And that goes back to that kind of tailored therapy design research aim, which is that if you could understand what truly drives colon cancer amidst all of this heterogeneity, you might be able to target that specific mechanism with a new therapy. And then, of course, because no two patients are the same, we can't have just one preclinical model, right? We can't have a single mouse to test all therapies. We're going to need to make preclinical models that are flexible with respect to genetics, so that we could define a driver in the setting of, some of these genes that are mutated in some patients, but that are normal in others. And so, to summarize, the past kind of 20 or so, or even longer, 30 years of research, we're limited in our ability to address that patient heterogeneity. So that's about 15 minutes or so of introduction, and I'll talk briefly about our efforts to address genetic commonalities, and that was also kind of what drove the interest by the folks at Forbes and for the 30 under 30 thing, and then I'll briefly address the preclinical models. So APC is a tumor suppressor. It turns off the Wnt signaling pathway. You could think of the Wnt signaling pathway as the engine to a car. If it is turned on, the car is moving. In this case, when Wnt is turned on, the cells are dividing. And you could think of APC as the brakes. And so when APC is engaged, the car comes to a stop and cells don't divide. And here's all of the, the brake lines and the engine and the motors and all that. These are the molecular circuitry of that system. But it's really simple to understand, actually, even though this graph is, or this diagram looks kind of complicated, because APC is the brakes, and when APC is broken, you pump the brake pedal, but nothing happens, and the car is going out of control. And we know that when you mutate APC, we have a familial condition in which every single patient with a mutation that's born with APC mutations will go on to develop polyps in their colon, and if left untreated, will develop into colon cancer. And then it seems like almost every single colon cancer we examine has a mutation either in APC or in other components of the pathway. So that would suggest that that the Wnt pathway, this entire system here, is really important to the disease. And so the question is, because we know that turning this system constitutively on, so losing the brakes or always turning on the gas pedal to the Wnt engine, is a hallmark of colon cancer, but no one's been able to address if we were to restore Wnt signaling back to normal, what would happen? And so in research in cancer and also in other diseases, we ask how do you model loss of a gene? And the first attempt actually sounds pretty crude, but you basically just added a mutagen to the mouse diet and you wait, and you just kept screening mice until they developed a disease that you were interested in studying. And this was work that's probably wouldn't be approved today, but decades ago that's how it was done. And so this is an old paper from the Dove lab, and they basically by chance discovered that when you give a mutation via mutagens in the diet, there are some mice that go on to develop intestinal tumors. These are actually tumors in their small intestine, and not the colon, which is different than what humans really get, but the fact remains the same. And it turns out when you look at the gene that's mutated, it's APC. So mice have, for the most part, the exact same genome as humans, and no different. They're the first gene that looks like it's lost in mice by random chance because of diet-induced mutagens is APC. And it turns out that this model, which was first described in the early 1990s, has now been published on about 320,000 times or so, right? So it's called the APC min model, and that is definitely the most ubiquitously available and widely published method to study colon cancer. But you already know something about the model that doesn't exactly jive with human disease, right? So we talked about how this gets small intestine tumors, not colon tumors, and we know it only has mutations in APC, so it actually doesn't get cancer at all. It has adenomas, that stage 0, which is a benign tumor. And finally, because it doesn't have any of the other dozens of mutations, it may not accurately model the response to therapy. So if you were to design a new therapy that you thought was going to cure colon cancer in humans, and you gave it to this mouse model, you would only be testing how it affects cells with an APC mutation, but not all the other ones that are going on in humans. A popular technique that has made headlines in the common press is called CRISPR-Cas9, and I won't talk about too much, but essentially it's a gigantic advance in our ability to target genes and cause specific mutations. So you can start with any cell of interest. You begin, in this case, with colon cells. You can engineer this Cas9 system to essentially cut out APC, right? So you can target these like molecular scissors right into the genome, and it cuts the DNA, and you're left with a deleted APC. And as a result, you ask, do the mice get tumors? And they do, and that was work that we performed in the first half of my PhD. So that's exciting now, because this can happen a lot faster than simply adding mutagens to the mouse diet and hoping for the best. And then finally, we have this technology called RNAi, or RNA interference. And that's really key, because it gives us the ability to transiently turn off a gene, but then turn it back on. So to explain it again, we'll start with normal cells. We'll begin. You could do this in any cell of interest. You could study brain cancer by modeling this in brain cells. You could study colon cancer by modeling this in colon cells in the mouse. And we know that APC gene codes the APC protein, and so these RNA interference molecules are able to actually interfere with that. So as a result, we turn on shRNAs that turn off the production of APC protein. And the end result is basically the same as if APC gene weren't there, except we haven't touched the gene at all, right? And we ask, okay, well, do colon cells turn into colon cancer cells? And do they have that hallmark of hyperactive Wnt signaling? And then we can ask that key question: if we then restored Wnt signaling in these established colon cancer cells, what would happen? And we can do that because these RNAi, again, don't actually interrupt the genes. We just turn off the shRNA, APC protein comes back up, Wnt signaling goes back down, and we ask that critical question: do the cells go back to normal? And this work was all done in collaboration with really talented faculty at Cornell, including Luke Dow, who's a professor of medicine and a very close friend and trusted mentor of mine. So huge credit goes to him. I'll skip this. I'll simply say that we devised a system to allow us to turn off APC in the colon, and we also added a genetic trick such that if the cells turned off APC, they would also glow green, just like a jellyfish glows green. In fact, the gene that allows a jellyfish to glow green is the same gene that we cloned into the colon to do this. And that just allows us to ask real quickly, has a cell turned off APC? And so this is a colonoscopy of a mouse in which we're turning off APC by this RNAi, and you can see the formation 8 weeks later of all of these colon polyps, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 or so, right? And if the system were working as we imagined, again, these tumors should be green, and they are. So that tells us that the reason why we're seeing tumors is because, in fact, these cells have lost APC. Okay, I'll skip this, but I'll simply say that the adenomas have all of the characteristics of the human disease. Again, loss of tissue architecture, uncontrollable cell division, loss of the differentiation of the cells, and tissue invasion. And so then we say, okay, in these established adenomas, what happens if we restore APC? And again, this is just documentation that we can restore the protein back to normal. So this is a colon cancer cell now, 4 days, and we're 4 days after we've completely turned back on the APC protein. And we confirm that with this technique called a Western blot. And as we'd expect, this is a plot of Wnt activity. So when we turn off APC, Wnt goes up through the roof, which is driving the colon cancer. And then we restore APC and Wnt goes back down, right? So we're modeling what happens when we turn colon cancer on, and we're asking when we restore APC, the Wnt pathway goes down, what happens in the mouse? So here's an example of two different mice, and here's a tumor. Here's two tumors in a different mouse. We restore APC, and we can follow that exact tumor by longitudinal colonoscopy, and you can see that the tumors are shrinking. In some case, these tumors are completely gone. So this mouse has absolutely no observable signs of disease. In other cases, the tumors shrink into like a little fibrous scar. If you were to examine this or biopsy this, you wouldn't find any cancer there. It looks kind of like if you got a cut on your hand and there's a little scar there, weeks later. So this is really like the mouse is repairing a wound in its colon. And again, if we look at that uncontrollable cell division here, any cell that's green and this tumor is dividing as a result of the brakes being off. APC's gone, Wnt signaling's up, but when we restore Wnt signaling, all of a sudden those cells are no longer dividing, right? So we've restored one of those hallmarks of the disease, which is uncontrollable cell division, and of course that would be great news for cancer patients. In this case, the cancer patients are mice, but if you were able to do this in humans, that would, we would predict it would slow the uncontrollable cell division of the tumor. And what's perhaps remarkable, and this is one of the reasons why this research kind of caught people's attention, is that not only were the mice cured of their cancers, but where the tumors used to be, 20 weeks later, there's completely normal mucosa, right? So it's not only that we were able to cure them of colon cancer, but the cells that remained behind went back to normal. And so that says that cancer cells have everything they need to go back to normal if you just had the right, molecular circuitry to reverse the changes that are caused by APC mutations. I'll blow through this really quickly and say that we engineered the additional second and third most common mutations in the humans into the mice. So KRAS and PT3 are the second and third most commonly mutated genes in human colon cancer. We gave those exact mutations to mice. They got terrible, terrible colon disease. And if we restore APC, even though they still have these two mutations, the cells go back to normal. And everything I just described to you holds true. And so the question is, what's happening? I'll kind of go through this real quick. We grew the colon cancer cells in plastic dishes in these three-dimensional gels that allow the cells to, under normal conditions, form miniature guts. And so what you're looking at here are cells that are harvested from a mouse, although you could do this from a man, the exact same way. And they grow into these little mini organs. And so what you're actually looking at right here is an intestine growing in a dish. And it has all of the features of a mammalian intestine, except it's growing in a plastic petri dish. And that it has stem cells that are undergoing stem cell homeostasis. It has enterocytes that are absorbing whatever it can because they're there to absorb, fats, sugars, minerals, proteins, etc. It even has cells that are creating mucus, and you can see the mucus in the center of these intestines. And again, we can assay or observe stem cell homeostasis in the colons of mice. This is a mouse colon that we've stained for cells that are dividing. And so these are those active stem cells that are proliferating up. And then we look at cells that are differentiated. Those are here are green. And what's interesting about the mouse organoid cultures, these mini guts, is that they completely recapitulate that stem cell homeostasis, right? So there are dividing cells at this out periphery, and then they migrate into the center of this mini gut, and they stop dividing, and they differentiate. And here are all the cells of the intestine. And so normal mini guts from mouse or man do this in the dish. But when we repress APC, what we see in the dish is essentially identical to what we see in the mammal, in that APC comes off, Wnt signaling goes up, and now we've lost stem cell homeostasis. Every single cell is dividing. There's uncontrollable cell division. There's loss of that tissue architecture, right? And in this system, we turn APC back on, and everything goes back to normal, right? So now we know outside of the mouse, if you just look at mucosal cells in a dish, they have the capacity to go from adenomas back to completely normal mucosa in just a couple days after we restore APC. And this is also true if the cells have mutations in KRAS and, for example, mutations in p53. Okay, so that's kind of like the main take home from the first part. How are we doing on time? We're at 4:45.
You know, Kevin, I think how much time do you, I know we have, I don't know, Sharon, if we've actually seen any questions come in yet, but we probably, if we could probably wrap up in about 5 minutes, that would be great to give time.
Awesome.
Okay, does that work?
Yep, yeah. So I'll simply say that what we did to do all this was you know, it told us a lot, but it was a lot of work. So all the data I just showed you really quickly took I think 6 years to finally finish. Let me just blow through some of this. But the point is that this work, in order to study that, took 5 or 6 years to do. And if we wanted to study APC restoration in the context of the no two patients are the same paradigm, it would be impossible, right? It's these patients are too genetically complex to study using mouse models by the traditional methods. The other thing is that our mice don't really progress to liver mets or lung mets, lung metastases. And so most of the genetically engineered mouse models really don't do that either. And finally, as I said earlier, most mouse models don't get colon disease. In fact, they don't even get colon cancer. They get small intestine adenomas. And so that kind of, during my thesis, was like a maybe a naive moment as a student. I said, well, that's kind of ridiculous. We're studying colon cancer in mice that don't get colon cancer, right? And so I'll kind of blow through this deck and just show this schematic right here. So the second half of my thesis and our work has been to make a better model. And essentially what we do is we use those tissues that grow in plastic dishes and we transplant them back into mice. Now that gives you the advantage of being able to make, say, 10 mice or 40 mice or 100 mice all in one day, rather than having to set up hundreds of breeding cages where litters are born invariably and then you can study 12 mice at once, etc. And we were able to show that we could get mice to actually progress to metastasis, which is key because this is the stage of cancer that is the most deadly in humans, and so we want to be able to model that in mice. And so perhaps I'll just stop there. But the second half was really technology, and we just published that method, and we're excited to see people slowly request reagents from us or technical advice from us on how to use this method, because they're starting to study their own questions in their labs using this new method to create colon cancers in mice. So that's probably a nice general overview of what we've been working on. I'm happy to take any questions.
Great, thank you so much. That's really quite interesting, and we still see your slides up. So
Yeah.
so that's fine. But we did have one question come in so far, and again, I can try to unmute you, the folks that are on the line, but let's just get through this one question that was typed in. Oh, so I believe in the beginning of the presentation you were referring to the cells as daughters. Why is that, and is that a common term that's used?
That's a good question. They could be sons or daughters, really. I think that's just kind of the nomenclature used in the field. Okay. So if whenever you're studying cells in like cell biology labs or molecular biology labs across the world, if you have a cell that divides into two different cells, those two cells are referred to as the daughters of that cell division. But there's no sex difference there. It's just a way to convey that it's the next generation of cells after that cell division. It's a good question, though.
Gotcha. So we did have a comment come through from Julie. Julie, I'm going to try to unmute you and see if you can maybe comment through the phone. Are you there?
Yeah, I'm here. I'm just overwhelmed.
Can
you hear me?
Yep, absolutely. Any thoughts specifically come to mind, or any clarification that you need?
Wow. No, I don't need any clarification. Boy, I just wish that if you turned back this is wishful thinking. You know, if you went back and you didn't have to create that these mice actually developed the cancer and that turned to carcinoma and then metastasized, and if you turned back that Win pathway or, you know, turned it off or on, there's just so much information here. I need to go back and think about it. I've been taking copious notes. It would be so wonderful if it was just that simple, but, you know, then he went on to show me that, no, it's not just that simple, and, you know, it's just kind of, I'm a stage 4 patient, and so, you know, you kind of go, wow, this is so exciting, and then, wow, we still have a ways to go. That's all. It's just a comment.
Yeah, well, thank you for the comment, Julie. It is like, if you could think of it like the most difficult puzzle in the world, in that the heterogeneity, meaning that the types of differences we see from patient to patient are like, you know, two different stars in the sky, and that's kind of been the focus of like understanding if there was one commonality that could be honed in on. Now, what we would predict is that if you had one drug, or this is not we, others in the field have predicted that if you only gave one drug, the cancers would eventually mutate a gene or genes to become resistant to that one drug. And so a lot of people imagine that the cure for, you know, things like colon cancer that have such a variety of mutations will almost demand that we use 2 or even 3 or 4 drugs all at once. And so now the question becomes, how do we figure out how to do a clinical trial with 3 or 4 drugs all at once? And that would be safe and perhaps effective in that trial. And that's what people are doing now.
You know, I guess it has to be frustrating to you, because I hear this a lot of time from lay people, and I'm a lay person as well. I was a nursing major before, so I consider myself maybe not so much lay, but, you know, you hear all the time that, oh, there's this magic drug out there, and the, you know, industry and big pharma and all of that are just making it so that cancer isn't cured because it's such a big money maker. And then you look at, you know, the genetics behind it, and you try to explain to these people that no, that's not really true. If you really understood what was going on at the cellular level, you'd know that that wasn't true. And this is just such an example of why that isn't true. I mean,
Absolutely.
It's phenomenal. What you've done is phenomenal, and you know, you guys are just incredibly bright to come up with all of this stuff. It's just amazing to me that, you know, that the progress that has been made, and that's the other comment that's often made, especially in colorectal cancer, is that, you know, nothing new has come out until just recently, the last couple of years, and, you know, mostly in cases of Lynch syndrome, where they've gotten the PDL and the PDL1 connection, and that's not the majority of cancer patients, but, you know, it's just phenomenal, amazing, and, you know, keep it up. I want to live to be cured.
Thank you. Thank you very much for all of your very kind comments. I can tell you that, you know, a major question is, okay, so for example, we know that PD-1 and PD-L1 therapies haven't shown incredible effects in sporadic colon cancers. So these are patients that don't have Lynch syndrome, right? And so the question would be, what drugs can we combine it with to make patients responsive? And to do that, you need a couple of very interesting things. So you need a model in which PD-1 therapy can have an effect. And it's interesting because, for example, a lot of labs will take human colon cancer cells and they'll inject them into a mouse that doesn't have an immune system. And you have to do it that way because if the mouse was fully immunocompetent, it would just reject the human cells, not unlike a virus or bacteria, or like, if you transplanted a kidney into a person that wasn't a match, they would reject it, right? That's the role of the immune system of the mouse or the human. So this model, one of the things I kind of really glazed over at the end, might be able to address that, in that we can build colon cancers in mice that have fully competent immune systems. And now that offers the platform to study how can we augment the immune system with drugs or therapies to have an effect on colon cancers in mice that kind of resemble the patients that, for example, don't have Lynch syndrome or are not microsatellite unstable, for example.
Dr. O'Rourke, we did have a comment from Lyra Chitwood as well come through. Lyra, I'm going to unmute you there.
Can you hear me?
Yes.
Yes, I can.
All right, Mike, I have a question. You mentioned that the WNT pathway is the predominant driver of stage 1 CRC. Is there a different pathway that's the predominant driver later, like in stage 4?
That's you could be my thesis advisor. That's the exact same question that we asked ourselves, and that we're continuing to work on now, and that's a critical one. So what I can tell you is that we took colon cancer cells, and this is the experiment we did. So we injected them into the liver of a mouse to model a stage 4 colon cancer. Now, in some ways, that model is artificial because these cells did not have to undergo all the steps that a truly authentic colon cancer metastasis had to undergo, right? So in humans, the cancer cells start in the mucosa of the colon, they invade the muscle, and then the lymph nodes, and then the vessels, and eventually to a secondary organ. But however, we skipped that process and just put them in the liver, and we restored APC. And what we observed was that the tumors underwent differentiation. So whereas there was normally uncontrollable cell division in these tumors, what we saw was these cells starting to form colon tissue in the liver. And
normal colon tissue.
Well, yeah, so that was pretty remarkable. So we saw that they were producing, for example, colon mucus in the fake glands that were arising in the liver. Now, that from a biological standpoint, I think is very interesting. To step back from patient care, that's an interesting leaf to add to the tree of knowledge, right? We did not know that you could grow colon tissue in the liver. But the other more important part for patients, it says that, let's say you had a therapy that was able to turn down what we think is still the predominant driver of stage 4 colon cancer, which is the WNT pathway. What you might get is colon mucin production in other organs or where you have metastasis. And what actually happened to the mice is, so for example, if you restore APC in a lung metastasis, you started to see mucus production in the lung, and that actually led to respiratory problems for the mice.
Yeah.
And so that's an interesting clinical correlate to the effect of this drug. So we're not 100% sure what that means for what a clinical trial would see or observe if you had a Wnt therapy in humans. But it does say that you ought to be looking for the signs of whatever may happen as a result of colon mucin production in other organs.
Okay, thank you.
Yeah, no problem.
All right. Well, Andy, are you still on?
Hi, everybody. Yes, Sharon, I have to run, but Kevin, I was staying on till the bitter end to listen to the final comments. So huge thanks. Sharon, I'll let you wrap it up completely about our next steps and the like. But Kevin, we can't thank you enough for all of this great work. And, I think the organization will continue to partner with you. And thank you for really donating this time for us today. Really appreciate it.
Well, pleasure's all mine. I'm very excited to be able to interact in this forum, and I look forward to all the things we're planning in the future.
So if anybody has any final questions or comments, again, on the right-hand side, you can type that in or raise your hand. I'll leave it open for about 10 more seconds since we're getting down to the end here. All right, Dr. O'Rourke, thank you so much again for taking the time. It was a really amazing presentation, and I know we all learned a lot. So thank you, and thank you to everyone, all the research advocates who joined us.
You're very welcome.
All right, have a great day.
Thanks for having me. All right, take care.
All right, bye-bye.